Mutation was first time studied by a scientist named Hugo de Veris. He studied this on a plant named evening primrose. He found several mutations in this plant, and then he defined mutation as a phenomenon which results in alternation of the base sequence in a DNA. So, he is going to change the entire base sequence. Now, how is it changed? It is changed due to mutagens. What are mutagens? Mutation causing factors. So, there are mainly two types, physical and chemical. Many kind of physical factors are there. I am just giving you one example, UV rays here, and chemical, say I give you as example as mustard gas. Now, when this change happens, we see that there are three types. So, how many types of mutation we see? There are three different types. First one is called as base substitution. So, base substitution is otherwise called as point mutation, and then you have deletion and you have insertion type. So, the first one, as we have discussed, we are going to discuss as point mutation. So, what are the types of point mutation we can expect? Two types of point mutation are transition and transversion. In case of transition, you see purine getting converted to another purine, or pyrimidine getting converted to another pyrimidine. So, let us quickly give one example here, or rather the only example. So, pyrimidine, you know, thymine is there and cytosine, so they get interconverted. Purine, adenine to guanine, it is interconverted. But in case of transversion, you see that purine is interchanged with pyrimidine. So, you have four possibilities here. So, the first possibility, say thymine getting converted into adenine and guanine, cytosine getting converted to again adenine and guanine, then you have adenine getting converted into thymine or cytosine, and guanine getting converted into thymine or cytosine. So, these are the two types of point mutation, but on the basis of point mutation, we have three separate categories. So, what are they? First one is silent mutation. What do you mean by silent? Keeping quiet, right? A mutation has occurred, but you just don't know how the mutation has occurred. So, I will give you an example. When you have a chain as AAA or AAG in DNA, what forms in RNA? Obviously, the synthesis would be UUU and UUC. I hope you remember or you know that both of them synthesize one type of amino acid, it is phenylalanine. So, what do you understand here? That if there is a mutation in the third position of the triplet codon, in most cases there is no difference and the same amino acid is synthesized. It depends on Wobble's hypothesis. But there is also another condition, what we call it as missense type of mutation, where we are misreading it, changing it. There is a replacement. Now, what happens in this case? I will give you a real-time example of sickle cell anemia. In case of sickle cell anemia, there should have been glutamic acid in the sixth position of beta chain of hemoglobin. You know, hemoglobin has 2 alpha and 2 beta chains, and in this beta chain the sixth position should be glutamic acid. But this is replaced by valine, because of which the person suffers from sickle cell anemia. But this is a 100% point mutation because there is no effect before or after this replacement. So 1 to 5 and 7 onwards, there is no change. There is a criteria 3 which is called as nonsense mutation. This is a very big trouble because here a stop codon is going to be formed. Okay, on the RNA lines, what are the three types of stop codon if you know? UAA, UAG and UGA. So, this is in RNA. So, let us form it in DNA. How would it be? You should remember that instead of uracil, there is thymine here. So, here will be ATT, then you have ATC and then you have ACT. So, this is in case of DNA. So, now assume there should be a chain which should be reading as GTC, TC, GTC, and one of the changes which has occurred here is just having a in between, or say this should have been TTC and this T has been replaced by A. So, again I repeat, it should be GTC, TTC, GTC, but it is replaced by A. When it is replaced by A, you can see here ATC becomes UAG as a stop codon and the chain stops here itself. The protein cannot synthesize here on. So, this is called as nonsense mutation. Now, I hope you are clear with base substitution. We are moving on to the second one, deletion. In case of deletion, say we are going to have an example here as ATT, CTC, GAT and so on. What happens? Assume suddenly I remove this one. Okay, let us have one more here. What should have been forming is, this should have formed into one of the codons, then the next one, and GAT should have been the next codon. But what happens now? There is a frame shift. It is shifting to ATT instead of GAT. So, what happens from here on? There is always a frame shift which is going to be in the right side. So, deletion brings about frame shift towards the right. So, what happens here is, unlike point mutation, the mutation is not restricted to one amino acid. Thereon, all the amino acids starts changing. Similarly, on the same lines, you have addition. Here, the frame shift is towards left. Let us keep the same example. So, I have ATT, CTC, GATT and so on, but I am making an extra addition of A here. So, what happens? Originally, it should be ATT, CTC, GAT and so on, but now I have ATT, then ACT, CGA and TT and so on. So, what is happening? The frame is shifting towards left side. So, these two conditions of deletion and addition comes under a bigger category called as frame shift mutation.